So now here we are. We've seen what Gregor Mendel did and how he made some assumptions about genes being in different forms called alleles, as well as the fact that offspring are mixes from both their parents, each parent contributing one copy of this information. Remember, he didn't know about chromosomes, DNA, or genes. He called them factors. And now that we've also gone through the process, the steps of meiosis, we also know now why it's random, which genes we pass on to our kids, and how and why we only pass on one of our two copies. Remember, in essence, it's really, as we had stated before, it's like flipping a coin. Same principle. But with that in mind, we've got a great trick up our sleeves. We don't need to go and do all of the genetic crosses like Mendel did and wait and see what the offspring come out to be and then cross those offspring to see what the offspring's offspring are and offspring. There's a much better trick for us to do that. We don't have to flip coins. We don't have to have thousands of kids. We can actually predict the likely outcomes of genetic crosses for any particular trait using what is called Punnett squares. Now, obviously, squares. How many squares do you see here? 1, 2, 3, 4. Oh, wait, there's a fifth one. 5. Pretty simple. We've got some letters mixed in here, and it may look very familiar to you from what you saw in 8th grade. Great. It's simple. It's effective. So let's take a look at how we would go about doing these, because I see some familiar concepts and terms. I see sex cells, male and female. I see letters here representing genes, but they are separated for some reason. So let's take a closer look at what this is, as well as how it works. So what we really want to focus on if we do this is how do we set up and do these actual Punnett squares? Because we have to remember a couple of things. First, and most importantly, remember, is that each and every single parent can only donate one copy of each and every gene, because remember, in meiosis, here are my beautiful chromosomes, our homologous pairs. I know you love the homologous pairs. Look at them. Ooh. Let's say that this one carries copies of genes right here that are a big T. This one carries a little t. Now, they're different versions, but remember, in meiosis, these two chromosomes during meiosis 1 separate or segregate. So our copy of a chromosome with a copy of a big T goes to one cell. The chromosome with the copy of a little t goes to another cell. Very, very, very important that we remember that. That is huge. Also, remember, it's random, thanks to meiosis, which allele they pass on through their gametes. They could pass on, as in our previous version, a big T or a little t. Totally random, like flipping a coin. So how do we actually do these Punnett squares with that in mind? They are simple. Simple, simple, simple. The first step in the process that we would really want to do and make sure is that we need to write down the genes of the parents. Now, what do we mean by that? Is, let's say, remember, each parent has 2 copies of a gene. So when we read through a problem, we always want to write down what are the parent's genes. From our previous screen, the example with the chromosomes, that parent, if they had those 2 chromosomes, would have 1 copy of the big T gene, 1 copy of the little t gene. We absolutely want to make sure we write that down. Secondly, another rule that we want to do is we've got to go back and determine what can they pass on to their kids. And that's simple enough. Remember, because of meiosis, we can pass on 1 copy. I could either pass on the big T in this case, or I can pass on the little t. Separate or segregate your alleles. Super simple. And the last fact is that we would then take our alleles that we're passing on and write them on the tops and sides of the squares. Each parent will go along one side of the square. So this parent right here can pass on, we'll stick them on this side of the square, either that big T, or our other option is to pass on a little t. There we go. Easy as pie. Once we know what the other parent is and their possible alleles, we can put them up here and here. And then it's simply a matter of filling in the boxes. If this parent gives a big T, this parent gives that gene, there's our 2 copies. That's what the kid would come out to be. And notice that we've got 4 options here within our square. Pretty nice. Sounds easy, right? Let's go ahead and look at one and try one, just to be safe. Filling in our boxes looks so easy, doesn't it? Simple, simple, simple. So as I said, if we try one, let's make up a fake example using anything. And we'll be focusing on humans a lot, so let's look at cats for right now. So let's suppose that in cats, there's a gene, and I'm making this up because I can do that because I'm Bentley and that's what we do. Weird teachers. Let's suppose in cats that there's a gene, and one gene only, that controls fur in cats. And it comes in two forms. As we stated here, the big F gene is the dominant gene, and that would cause the cat to have large, fluffy fur. Draw a quick cat here. Here's my cat. There's the little nose. Oh boy. I hate cats, actually. And here's his fluffy fur. Oh, he's so fluffy. He's awesome. And okay, whatever, Bentley, stop. But let's say that we have two cats mating. One parent, we'll put a little X here to denote they're being crossed, like Mendel's pea plants. And one of our cats, as we can see here, is big F, little f. His two copies are big F and little f, the two copies of his furry gene. And let's pretend the other cat is the same thing. So there are our parents. We did our step one. We've written down what their genetic makeup is. Now remember, step two, we want to figure out what can they pass on to their kids. Keeping in mind, these alleles segregate. So this parent can give either a big F or could pass along a little f. Same thing with this parent. Big F or could pass along a little f. Easy enough. Now all we have to do is set up our square so we can do this little crazy mix-up of different varieties, different outcomes. Let's put this parent right here. We'll put him on this. Actually, you know what? Let's put this parent on the top. Do you think it matters if that parent's alleles go here or here? It doesn't make a difference. 3 times 4 is 12. 4 times 3 is 12. Order is insignificant. This parent up here we'll put on the sides. They can also give a big F or a little f. Now all we do is slide in and fill in our boxes, as we said for number 4. If each parent gives a big F, the kid would come out big F, which would make that kid, that kitten, that cat, have fluffy fur. Big F over here, little f down here. What would that make the kid? Well, he's got two different genes here, but the big F is dominant, so he would also have fluffy fur. Same thing over here, big F with a little f. Notice that it's a good practice to always put the dominant letter first to keep track of what the genotype will be, is fluffy. But notice here, this is kind of weird. Both parents up here had fluffy fur, but what if each parent gives a little f and a little f? This guy fills in there, this guy fills in there. There's our little f, and that kid would end up having matted fur. Wouldn't be fluffy. Look like a wet cat. So they stand, if we look at this now, there's a 3 out of 4 chance of the kid having fluffy fur, a 1 out of 4 chance of having matted fur. Wow, that looks so easy and predicts the future. Now notice, each time we have a kid, that kid could come out as any of these outcomes. This is just showing us the possible outcomes, not what each child will be. In other words, if we had 4 cats, one wouldn't be this, one wouldn't be that, one wouldn't be that, one wouldn't be that. That's not a guarantee. Each offspring could be any of these 4. Kind of crazy. Makes sense? Now, just really quickly here, some useful terms that we need to know that we're already somewhat familiar with. Dominant or recessive. When one allele overpowers or hides the other, like in our last cat case, fluffy fur was dominant, the fluffy fur will always show up. Even if in that individual's genes they have a recessive trait, recessive allele, the dominant trait will show up. Recessive, in essence, is kind of hidden or masked. The only way we could show that recessive trait was like in our last example if we have 2 little f, 2 recessive alleles. Make sure we know these 2 terms, very important. Also, we need to know the difference between genotype. What type of genes do you have? And here are some examples. I could be big T, little t, little t, little t. What are my 2 copies, in essence, my makeup of my genes? Because the genes, genotype, determine physical phenotype, the physical trait, like are you tall or short, black or white, purple or white, fluffy fur or matted fur, that kind of thing. Make sure we know these two. We will always need to know these whenever we solve problems. And the last two terms we need to know is the difference between homozygous, zygote meaning when we join two sex cells together, so we mix our two alleles together here. Homo means same. So if for a gene you have two identical alleles, little a, big T, etc., you would be homozygous for that trait. Heterozygous, hetero meaning different, is when your genotype contains two different alleles. One thing to be careful of here, notice that big T and big T little t are different genotypes, but they would both show the same dominant phenotype. Be very careful of that. So now one last little trick there, now that I think we're able to do what are called monohybrid crosses. When we look at four squares, we can actually track two traits at once. Two traits would be dihybrid. And since we're looking at two letters, two genes, two traits in this case, it's not going to be a four square. It's going to end up being, holy cow, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 16 squares. And notice in each gamete, they're donating one copy of each gene, an R gene and a Y gene. So how are we going to do that? Well, the method is actually, we're in luck, is the same. Write down the parents, determine what they can pass on, fill in the squares. But there's a catch, remember, that once we determine, the hard part is going to be determining which genes they pass on. Remember, we can give one copy of each gene, so we're going to have to employ what's called the FOIL method here. Now, the FOIL method is actually, just like in math, is pretty darn simple if we look at it. I can give one of these A genes, but also at the same time one of these R genes. So what we want to do is F our first. Go with our first options. I could give a big A and a big R. That's one option what I can pass on. O for outside. I could pass on that A with that R, my two outside alleles. There's another option. Inside alleles right here. Little a, big R. Or L for last. My last A and my last R. There's my other option. So notice that I have four different options here that as a parent I could pass on if this were my genotype. Remember, I give one copy of each gene and it's totally random. We can't forget that. All right, well, so what? So now how do we do it? Pretty simple. Write out the parents' genotypes, write out what they pass on, one copy of each gene, and we fill in the squares. Woo! Now here's an example back with cats again. But if you notice, look at these two parents, our two parents that we are mating here. This guy crossed with this guy. Each parent has to give an S. Each parent has to give a B. So if we FOIL this guy, he could give a big S with a big B. That's right there. Big S with a little b. That's right there. Little s with a little b. There it is. And little s with a little b. Bang. Same thing for this parent. And look at what we have. We have 16 options, but what's strange here is for these traits, we're actually studying their tail length and their coat color. Notice up here, big B is dominant for brown over little b, which is white. And short tails, the big S, is dominant over long tails, a little s. So if we look at this, all we got to do is count them up. If we cross these two brown short-tailed cats, notice they're heterozygous for each, we would end up having 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 cats that were brown, but they're not going to be the same. Some of them will have short tails and be homozygous. Some will have short tails and be heterozygous. Wow. Some of them are even going to have long tails. Short tail homozygous. By the way, sorry, I screwed that up. Short tail heterozygous. Long tail homozygous. Holy crap. And this is homozygous brown, heterozygous brown. This guy's white because he has two recessive traits. Wow. A lot of diversity, but it's the same way no matter what when we analyze Punnett squares. the same no matter what we're doing.